This Week in Microbiology is brought to you by microbe.tv and is supported by the American Society for Microbiology. To learn more about microbes, visit microbe.tv and asm.org. This is TWiM, This Week in Microbiology, episode 330, recorded on April 3, 2025. I'm Vincent Racaniello, and you're listening to the podcast that explores unseen life on Earth. Joining me today from Ann Arbor, Michigan, Michelle Swanson.
Hello, good to be with you again.
How's Ann Arbor? It's all good? No, it's not.
Let's see how the others are doing.
Okay. Also joining us from South Carolina, Michael Schmidt.
Hello, everyone. We don't quite have Chamber of Commerce weather, but the azaleas are in full bloom. Spring has sprung. Everybody's car is a mild form of yellow to green.
Yes.
Pollen. And my nose is running like a drain. So, must be spring.
Well, it is 20 C here. And if you know the Celsius scale, that is 68 Fahrenheit.
Yes.
It's one of those numbers you can easily convert. Also joining us from St. Louis, Petra Levin.
It's good to be here. It is definitely not Chamber of Commerce weather, but it is very humid. So that's a prelude to our spring. It's about 57 Fahrenheit. So I don't know, 18 centigrade. And yeah, I would say the weather here in the spring is about as predictable as everything else has been lately.
I mean, unpredictable.
Yes.
Yeah, we had a 2-inch rain last night. It was amazing.
That happens usually at least once every week in the spring in Missouri, but yeah.
Wow.
If you enjoy these programs, we'd love to have your support to continue them. We don't do ads, if you noticed, to disrupt your learning. So please support us. We're a nonprofit, so you can, in the U.S., it would be federal U.S. tax deductible, microbe.tv/contribute. Today, I am going to do a snippet. I've been meaning to do this snippet for weeks.
It's very cool.
And it is it's a very complicated paper, which it isn't helped by the writing. The writing is a bit obscure. They assume that you know a lot. Anyway, it's a Nature article. It's called "Engineering a Genomically Recoded Organism with One-Stop Codon."
And it's ochre.
It's called ochre. They make this new bacteria called ochre.
Yes. What are the three? It's ochre, amber. And
umber.
opal.
Opal? Not umber? Opal, amber, and...
I can't... Yeah, these are very old school names for these. Like
UAA, UAG, and UGA, right?
Yeah, the stop codons. So I like that throwback here.
Anyway, mostly from Yale University. And we have 3 second authors and one first author, Michael Grome, and 2 senior authors, Jesse Reinhardt and Farron Isaacs. All right, so as you may know, the genetic code, 64 codons, right? And there are 20 amino acids, so it's degenerate. Some amino acids have more than one codon. And there are also stop codons to stop translation, UAA, UAG, and UGA. And a while ago, in fact, in E. coli, so these are called genomically recoded organisms, GROs, right? Not GMO, GRO. But if you put it in your cheese, it'll be a GMO. A while ago, someone made an E. coli where all the TAG stop codons were replaced with TAA codons. So they replaced one stop coding with another codon. And, you know, when translation stops, the codon is recognized by release factors. There are 2, ERF1 and 2, RF1 and 2. I'm thinking of eukaryotes. I put the E in front of it. And the translation stops. And there sometimes tRNAs recognize a stop codon and they stick a tRNA in. But what they did, they recoded all the TAG stop codons and also deleted the gene for RF1. So now that codon is free. They say, we released the codon.
Is that like
To the genetic
releasing the
engineers
Kraken?
everywhere.
It's so funny. They say all the time, we released it for reassignment. It's like it's the
Yeah.
military.
Exactly.
So what does it mean? It means you could have it code for some other amino acid if you wanted to, because it's no longer involved in termination. And they think, well, there are 3-stop codons, so let's take out the other one and just have an organism with 1-stop codon, and then we could use the other 2 for making unusual proteins and so forth. Okay, so that's what they do. They call it compressing a redundant codon functionality into a single codon, because it's redundant. 3-stop codons is redundant, right? So they're going to make an organism ochre with just UAA as the sole stop codon.
But it takes a few more steps, as they found out.
Oh my God, this thing is just amazing what they have to do. All right, so they start with the E. coli that's where all the TAG codons have been removed, right? That's a good start. And so how many TGAs are there?
Almost
So
1,200.
a lot, 1,000, over 1,200 TGAs.
In simple E. coli.
In E. coli. And so they take them all out.
So one question I have, by virtue of the fact that they took out the TAG codon or UAG codon, is what happens to selenocysteine, which is an amino acid that's used in a very important enzyme in E. coli, formate dehydrogenase. And many of the enzymes that use selenium in them, they use the opal codon to effectively put the selenocysteine in. And because it's inefficient putting it in, that's why you don't have selenocysteine in all of your amino acids in those instances where you need it.
So in fact, those genes that use selenocysteine, they left them alone.
Okay.
They didn't change the stop codon, right? Yeah, and in fact, there were the enzymes you say, formate something.
Formate dehydrogenase and formate hydrogenolase.
Yeah, I think there are 3 or 4 instances, and they left them alone. Yeah, but they have to do major mutagenesis, but they have a process called MAGE, multiplex automated genome engineering. So you could do thousands and thousands of changes at a time. This is George Church kind of molecular biology. George Church, a Harvard scientist who just develops ways to do things at huge scale. And that is what this is all about. So they mutate all of those. Yes, and here we go. 3 formate dehydrogenase genes were not containing internal TGAs, right? We're not targeted because they need selenocysteine. And they change all of these cycles of this MAGE business, and they can get rid of all of them. And they have a strain which lacks all of them. But what's interesting is in the process of doing this MAGE, they accumulate 700 unintended background mutations.
Oof.
Not surprising, right? Yeah, because that happens all the time. When you mutate one thing, something else.
But you don't like to think about it. We like to be in denial about that.
Absolutely.
Yeah, you don't sequence your strains, but when you're making this many, you got
to. But what they did,
you're putting in a lot of even getting in, changing anything, you're going to just have to get some mutation somewhere else, just the way mutation.
So what they did was they made sure they grew well.
Oh, that's good. Fitness. They used fitness as their criteria. They would
take the fastest growing strains and take them on to the next step. But
as
in the
if
end,
that's not selecting for some.
No, it's not. But they didn't want to sequence all of this stuff. All right, so now determination, the release factors RF1 and 2, which they recognize the stop codons. They themselves look like a tRNA, and they cause the peptide to be released, the protein to be released, and terminate translation. So there's 2. RF1 terminates at UAG and UAA, and RF2 terminates at UGA and UAA. So there's some overlap, right? So RF1 is already gone, but RF2 has other functions beyond termination, so you can't just delete it. And anyway, you do need to have some termination still, right? So they did a whole project on its own, identifying mutations, amino acid changes in RF2 that would allow it to perform these other functions, but not allow it to terminate at UGA. Okay, they attenuates termination. They don't abolish it completely, but if you have UGA present, which is what's going to be in this strain, just UGA, then this factor is going to keep recognizing it, and it's going to compete for whatever you want to amino acids you want to put in. So you want to reduce it as much as possible. So there's an enormous amount of mutagenesis. They do structural prediction using AlphaFold of RF2, and they identify some potential amino acid changes. And they identify one at a specific amino acid, R170K, that seems to selectively impair recognition of the stop codon, but not other functions. And so they do a bunch of assays to measure what's going on. And they find that in the mutant where this RF2 has been altered, they have 42-fold enhanced read-through relative to no suppressors. So UGA is attenuated compared with the wild type RF2. It's substantially attenuated. So they want to move forward with that.
And can I add, they have a really clever tool to quantify the read-through, and that is a fluorescence reporter where you have like a green fluorescent protein and a yellow fluorescent protein, for example. And then you put the stop in between, and then you can just plate out the cells and see how much red and green we get. So pretty clever.
This is full of clever assays. Yeah.
And there are 18 authors on this paper. So it was a lot of really detailed structural and biochemical work, but it was a team.
All right. So now they have this strain. But the problem is they do mass spec and they see that they're getting a lot of TRIP suppression. In other words, the TRIP tRNA is reading the one codon at UGA. So they don't like that. They don't want to have competition with TRIP tRNA. And so they modify the tRNA gene to abolish UGA.
As you would.
They abolish UGA recognition. And now TRIP is still encoded by another amino acid. I forget what it is, but it won't be put in at the stop codon. I mean, all these things you had to do, right? It's not just wiping out the stop codon.
Right. Who knew? I mean, when we draw out transcription and translation, it's schematically, it looks very straightforward.
Yeah. So one base change in the TRIP tRNA eliminates this decoding at UGA, and now UGA is free for reassignment, as they say. All right, now the problem is this strain, which in a moment we're going to show you that it actually works, has horrible fitness. Doubling times are reduced by 50%. And it's mostly this RF2 change that is doing that. And so they say we're going to have to do further tuning to get this to grow better. Because, if you want to make a product, a protein with unusual amino acids, it can't be growing half as well. It's going to be a real problem.
And that's their motivation, is application and engineering.
Tuning. They want to learn how to tune.
All right, so now you have UAG and UGA freed up. And so they construct fluorescent reporter plasmids to insert fluorescent molecules at the two stop codons, two different fluorescent molecules, and so they can measure them, right? So they have to not only make a tRNA coupled to these molecules, but they have to also make the tRNA synthetase, right? Because that's the enzyme that's going to couple these chemicals to the tRNA. So they have to modify both. Well, a lot of things going on here. It's amazing. Anyway, it works. They can get both of these. They can put different amino acids at both of these stop codons that they've reassessed. And there's no more competition with suppressor tRNAs. So now they have this strain ochre. All the genomic stop codons are replaced with TAA. So in other words, all the TGA and the other one are replaced with TAA. So wherever you need a stop codon, it's now TAA. And then they engineered tRNA TRIP and RF2 so it wouldn't recognize UGA, one of the ones they want to liberate. And this experiment where they incorporated non-standard amino acids over 99% accuracy. So that's really good. And so, aside from the fitness problem, which maybe they can fix, now you have you have compressed termination to one stop codon, which is really amazing, right?
It is really impressive. I'm also, though, a little confused as to, well, first of all, that fitness defect is unclear why it has it. Are they dying, the cells? Are they just growing slowly? Are they activating some kind of like the stringent response where they think they're starving for amino acids?
Or the messages are simply turning over before there's really insufficient TAAs, the charged tRNAs to fill the stop codon that they're swapping.
That seems fixable, though. Like, it feels like if you just look for suppressors that grow faster, you're going to have more problems than if you go through the different options. And also, it's pretty straightforward. And the other question is, they kind of vaguely touch on what these might be useful for, but what are the practical applications of this kind of strain? I mean, I guess you can make proteins, like if you're going to induce proteins and purify them, or is it to study? I mean, for engineering, obviously, they probably want to make stuff.
Yeah, well, there are a couple of applications. They say, so make new kinds of proteins, right? Because you can have now lots of you could put these codons anywhere you want, right?
Right. So you'd want to make a protein with a specialized amino acid that you express at high levels. So they really have to get that fitness fixed. Yeah.
So that's what, but they also talk about safeguarding genetically modified organisms with enhanced genetic isolation. So if you have a special organism that you've made, you make it dependent on just one stop codon. I don't know how that.
Well, this reminds me of years ago when they were worried about genetically modified microbes. You effectively develop suicide genes that without the antidote, the microorganism would die, and that antidote was supplied on a plasmid. And when the plasmid was lost, the genetically modified microbe would just die because the suicide gene would be expressed because the antidote was on the plasmid that was expressing.
As far as I can see, this is self-contained, or ochre is self-contained. And so I don't know how it's biocontainment works. They don't explain it. I mean,
it doesn't seem like this guy is going to take over the world either.
No.
If it grows at like 80 minutes. I mean, I think the idea of like using it to express proteins with different tags on that, specialized proteins. I was just trying to can we use this to like tag a protein in the cell in a special way to see who it's interacting with or something in a more practical molecular biology, which I'm sure they're not thinking about. But.
Well, if you can model the interaction and make predictions and then test them directly using this.
Especially now given AlphaFold's ubiquity. I mean, I was at the South Carolina branch meeting this past weekend, and the number of undergraduates using AlphaFold to do experiments is phenomenal. I mean, their posters had AlphaFold in it, and it was incredible.
Well, AI is popping up everywhere.
Yeah, it's like
when you do a Google search now, it's all AI driven.
I know. I've had to get rid of Google search.
And I feel uncomfortable because I don't trust it.
I can't tell you how many posters I see like at our program retreats where even grad students put up AlphaFold and they're like weird, giant, unfolded domains. It's like, that's not normal to have your protein It's like, unless you understand what this AlphaFold is telling you and can justify using it in your poster, it's better not to use it.
Yes.
Because it is just imaginary at this point.
I heard today that all of the presidential orders, what are they called?
Executive orders.
They're written by ChatGPT.
I mean, all it is pattern recognition to the nth degree here.
I hope they check it over before they release it.
Let's get back to this explicit paper.
I think it's really exciting.
That's the paper. It's pretty cool.
Yeah.
But this is not something anybody can do. It takes a lot of
manpower.
Technology. But hopefully they
design this, it can be available to the field.
Yeah, make it available to everyone.
That's right. And the work, and since there's been a lot in the press about this research and who funds what, they got funding from DARPA, which is a federal agency, the National Institutes of Health, also the National Science Foundation, and then a couple of private foundations, the Beckman Foundation and the Carlsberg Foundation. So a lot of support for this pretty ambitious creative project.
It is really cool, and I think it just has a lot of power, but the question is,
what do you do with it?
What do you do with it? Yeah.
Well, I think now that we'll see, people start using it in some way.
Yeah, and I do want to understand that fitness.
Yeah, it depends on what new modified amino acids you can now pop into that released site.
Yeah.
Right.
But the unanticipated result is if we can figure out the fitness defect and how to tinker with fitness, that would have great utility.
I agree.
That is
They're working
doable.
on it.
I would love to understand that fitness defect. That sounds really exciting.
All right, now we're going to jump into the mouth with Michael Schmidt.
That is true. So I've been deep in the weeds. We're having our every 7-year/8 year because of COVID dental accreditation. So when this paper popped up, I thought this would be nice. It's entitled Identification of Vionella parvula and Streptococcus gordoni adhesions mediating co-aggregation and its impact on the physiology and mixed biofilm structure. This paper was published in MBio in December of 2024, and it has a variety of authors, and I apologize in advance if my French accent. It's Dorison, Bacon, Martin Galchos, Chamorro Rodriguez, Ventrenko, Quazaro, Villa, Deschamps, Briandet, Grabaldo, Gigot, and Boulogne. And so this paper, as you might think about, it's all about dental plaque. That stuff that requires us to visit our dentist every 6 months. So dental plaque is a polymicrobial community where biofilm formation and co-aggregation, which are operationally defined in this instance as the ability of one species of bacterium to bind to another species of bacterium. And this behavior plays a major role in the development of a very complicated organized consortium that dentists know as plaque. Why this matters is that dental plaque is, again, a polymicrobial biofilm where when we disrupt the nature of this biofilm or through perturbation, it can lead to an accelerated or decelerated development of dental caries, which are cavities no little kid wants, gingivitis, which is really the accumulation of plaque and tartar, which is, if you will, a mild form of gum disease, principally evident when you brush your teeth and you experience your gum is red, swollen, it bleeds, especially when you poke at it with the toothbrush or you floss. And left uncorrected, gingivitis can progress to periodontitis, which is a more severe form of gum disease where you have substantial inflammation, where you actually have bone reabsorption. So your jaw is actually dissolving that supports your teeth. And then your teeth become infected because you develop deep pockets, which you might have heard as getting long in the tooth, which is literally where you have gum recession. And that's where long in the tooth came from. As people's gums receded back into their skull, they would have these very big teeth. And the final thing you develop in periodontitis is you develop bad breath. Now, I'm not talking mild garlic breath or onion breath. I'm talking an odor that can peel paint from walls. And if you don't think I'm serious, I have patients who one of our periodontists says the wife comes in, complains her husband has breath that will literally take the paint off their walls. So one prominent member of this dental plaque community is a strictly anaerobic diderm, Vionella parvula. Now, diderm means it's gram negative. But if you look at what family it is in, it should really be a gram positive. But this coccus-shaped bacteria has actually two membranes, if you will. It's got the classic unit membrane that all bacteria have, and then it has an outer membrane that gram negatives has. It's a normal constituent of our oral flora, but when it gets into the wrong place, it can be associated with diseases such as periodontitis, dental caries, as well as various systemic infections, including the evil ones, osteomyelitis, and of course, meningitis. But this microbe is considered a bridging species. By that, I mean the Veillonella contribute to the formation of dental plaque by binding to the initial colonizers of the tooth surface, the first guys that are attaching to the pellicle, which is that substance that coats your teeth. The minute you stop brushing, the pellicle reestablishes itself. And it's a protein layer that serves as a protective layer for your enamel. So the Veillonella, being a bridging species, then recruits early and late colonizers to attach to it to help form this complex biofilm. And many of these organisms that are attaching to this growing biofilm can be pathogens like Porphyromonas gingivalis, Treponema denticola, which is a gram-negative spirochete, and of course, Tannerella forsythia, which is another gram-negative anaerobic rod that used to be called Bacteroides. And Porphyromonas, you'll recall from one of our previous TWiMs a few years ago, was one of the microbes that was implicated in triggering the development of plaque in the brain, where it was an indicator of early onset Alzheimer's. And all of these things come together and they form what an investigator termed, or the workers in the field named after this worker, they referred to this as a Sokransky complex. If you happen to follow the hierarchy of dental plaque, Sokransky published this paper in 1998, and I've dropped it into the show notes along with a couple of other papers, along with a stylized image of how workers in the field have showing who's sticking to whom and what's actually going on. And it really helps you understand why these authors did what they did. So Veillonella should be considered this ecological keystone species as being, if you will, essential for this oral biofilm formation. Now, the neat trick about Veillonella is its growth depends on lactate. And if you know anything about the founders in your biofilm's mouth, the streptococci, they're either principally homo or heterofermentive streptococci. That means their principal waste product is lactic acid. So the Veillonella are actually eating the streptococci's waste, which is lactic acid. And lactic acid is responsible for the dissolution of your hydroxyapatite crystalline lattice, which you know as your tooth. And that's how you develop cavities. So if you don't have enough Veillonella in your mouth, you're going to be more prone to cavities. So our understanding of how Veillonella parvula co-aggregates and then the impact of this aggregation has been long. There's been a quest trying to understand how to keep these good guys in there without displacing them with bad guys. And principally, that's was hampered due to the lack of appropriate genetic tools. Enter these workers. In this paper, Dorison and colleagues from the Institut Pasteur investigated this co-aggregation phenomenon using a naturally competent strain, which means they can do genetics, of Veillonella parvula, and the strain number is SKV38.
And if I could interrupt, Michael,
sure.
The first author, Louis Dorison, wanted to highlight Simonetta Gribaldo, who's also at the Pasteur, and he says she might be the only person in the world doing genetics on the Veillonella, and they were very fortunate to be able to collaborate with her.
So, as Michelle just said, they had a system where they could do genetics. Now, previously, the same group described 9 genes coding for trimeric autotransport adhesins, or TAAs, but not the stop codon Vincent just talked about. This TAA is trimeric auto-transport adhesion. So I guess this TWiM could be named TAAs. So the TAA in this case is a large fibrous homotrimeric cell surface protein that contains a modular domain that facilitates attachment to a variety of surfaces, as well as they can mediate this auto-aggregation, which literally is the clumping of the same species of microbes. Now, one of these auto-aggregating proteins, HAG1, mediates adhesion to oral bacteria and buccal cells. Buccal cells are the flat cells that line the inside of your cheeks and lips. They're the great source of DNA, and some of you may have even used them to conduct genetic testing on yourself when you sent off your swab to the now unfortunately bankrupt 23andMe or Ancestry.com.
Yeah,
so,
these are the cells they use when they do look for X inactivation.
Yes.
They can stain it. And this is like something we did. I don't know if they still do, but we'd take them and then we'd stain them, because if you're born with two X chromosomes, you only need one. And in the stain, you can see this little dot, which is the curled up X inactivated one. They're really cool.
It's really cool. And they're easy to liberate. Just a swab will do it. So back to the paper. These 9 TAAs, and they're named VTA, VTAA through L, and these 3 classical monomeric autotransporters are named VMA through C, A through C. And both VTAA and a gene cluster coding for 8 of these trimeric, homotrimeric surface proteins were shown to be important for surface adhesion and biofilm formation. In addition, VTAA mediates the autoaggregation of Vianella parvula. It means it's able to stick to itself. Now, the prototype of this protein family is from another diderm, Yersinia, specifically the adhesion YAD-A, which also has a variety of functions such as binding to collagen and other extracellular matrix proteins. But in this case, Vianella's VTA doesn't enable it to stick to our stuff. It just sticks to other bacteria. Now, Dorson and colleagues set out to identify the adhesins mediating co-aggregation of Vianella and Streptococcus gordoni while addressing how co-aggregation of these microbes impact the physiology in this mixed biofilm structure. Now, go back to the big picture. Why do you do this? And the reason you do this is there are people who make lots of cavities and there are people that don't make any cavities. So if you can figure out the right distribution, and when you look at that one figure that I dumped into the show notes, you can see that this is a really complicated story. So what you want to be able to do is follow this brilliant and elegant set of experiments where they actually follow the theme of how do you know and or how would you show, given they have a genetic system in which they can do binary or knockout experiments. For example, consider the observation that Vianella parvula uses specific adhesins to interact with Streptococcus oralis, Gordonia, and an Actinomyces auris, the so-called, if you will, base layer of the plaque from which other bad-acting microbes stack on top of itself. And so as they try to identify the potential ligands or the Vianella adhesin, they use their model microbe, which is naturally competent, and so they could do the genetics, and they performed an assay where they literally had separate cultures of this strain, SKV38, and it's grown in an enriched medium, which looks like LB, but the only thing different about this SK medium is the carbon source, in addition to having yeast extract in there, the carbon source, where it's at 10 milligrams per liter, 10 milliliters per liter, is sodium D-lactate or lactic acid. So it's growing in a medium with 10 mils per liter of lactic acid. And so they asked the question, does it auto-aggregate? And that was in one of their supplemental figures, and it doesn't. But it's capable of supporting these in vitro biofilms with similar diversity to healthy subgingival inoculum. So the medium in concert with the, if you will, the strain, the naturally competent strain, enables them to do the sort of experiments that they do. And the experiments, the way they display the data is really quite elegant. Recall that many of the microbes within the oral microbiome are very fastidious. So when SKV38 is grown on the medium, it doesn't stick to itself. And what they found is that it only co-aggregated or stuck to several species of Strep gordoni, Streptococcus oralis, one particular strain, and then one particular strain of Actinomyces oralis. It's a cool little figure, and if you look in the panel, the first figure, the smaller the bar, the greater the aggregation. So it's brilliant. You just look to the where the bar is the shortest, and you can see who's sticking to what. And they also found that it did not co-aggregate with Strepmitis, Streptococcus parasanguinis, Fusobacterium nucleatum, which is at the top of the biofilm. So it didn't stick to that. And Strep mutans, which is really what we teach the dental students. It's the first organism that colonizes the pellicle. And it didn't like sticking to that. It always was very weakly sticking to Strep mutans, one particular strain. And the figures are all the same. So it makes dissecting what they're learning very easy. Now, again, so here's where their procession of a strain that they could do genetics comes in really handy. Here they went about identifying which of the 12 Vionella adhesins were involved in the co-aggregation with these different partners. Now, they had previously constructed single deletion mutants of each of their adhesions, and again, performing co-aggregation assays through the mixing of independent cultures. So they're literally mixing, pouring things together. They were able to test these 3 strains, and they have aggregation buffers, so they can actually literally see them sticking together in a visual assay. This takes us to panel B of the first figure, and it really is all depicted best in figure 1F, where you can literally see this co-aggregation after co-culture between the Gorgoni and the Parvula, as well as the different adhesion mutants. The next set of experiments took us away from the oral cavity and dental plaque, where bacteria are adhering to other bacteria, but their curiosity got them wondering if it's like the Yersinia, where its adhesins can stick to us. And the answer was no. And again, through deleting individual or multiple TAA genes, Dorson and his colleagues showed that VTA also mediates co-aggregation with oralis, whereas VTA A and VTA E are involved in co-aggregation with Actinomyces auris, and VTA E and VTA D are needed for co-aggregation with Gordoni. So you have 2 separate proteins that are literally stuck out of this gram-negative coccus. So it's just sticking out of this, and they don't appear to mediate adhesion to host cells, in contrast to the TAA of the Vionella that we saw with HAG1, which is commonly associated with another species of Vionella, which is Vionella atypica, which is encoded by the positional ortholog of VTA-A, but is structurally distinct from VTA-A. So the nomenclature gets a little bit, you almost have to create your own cheat sheet to figure out who's sticking to what, and in order to keep it straight. But what's the remarkable bit to me was not that Gordonia and Vionella led to biofilm formation, but rather the deletion studies offered that when co-aggregation was prevented through either the deletion of VTA and Strep Gordoni or VTA-DE in Vionella, the resulting biofilms ended up segregated. So this is the big leap here, folks. They're understanding how we can disrupt dental plaque so we don't make this big complicated structure. And so there's an order, and metabolism is probably the driving force. Now they did a lot of transcriptomics on this that you guys can look at because it's a wonderful open source paper, and you can see all the transcriptomics, which effectively confirms the earlier experiments, and they really wanted to demonstrate that in spades. But they finished things off with a model where, and this is in figure 7 of the published paper, where effectively the figure offers us insight into the molecular determinants at the origin of co-aggregation mechanisms, or if you will, biofilm development between parvula and different members of dental plaque. They've identified these 3 Vionella parvula proteins and 1 new strep gordoni adhesion involved in this co-aggregation. And then through their transcriptomic studies, they address the impact that such co-aggregation on partner physiology and co-biofilm structure might have. So they're leaving us hanging a little bit, but it's enough for us to really get a sense of excitement when you read it. And they conclude they're able to show that Vionella uses specific sets of these multiple trimeric autotransports to specifically interact with other members of oral dental plaque. And while these adhesive capacities are not necessary, and we haven't invoked quorum sensing yet, because we know biofilms like to talk amongst themselves. And so we have a scam call, even though I'm in do not disturb. So while these adhesive capacities are not necessary for intracellular communication or quorum sensing under their condition tested, we can appreciate that the reduced distance between members of the biofilm using these adhesion molecules. So as you might expect, as the quorum molecules, which are nothing more than grenades thrown across the side for others to interact with, as they are released, the likelihood of encountering and then acting on a genetic switch that quorum factors do is likely more likely. So the concentration of the quorum factor, my prediction would be likely less or might be more depending upon who's stealing the quorum factors. Finally, such co-aggregation phenomena are likely to contribute to this highly organized process of dental plaque formation by modulating the successive addition of interacting bacterial species, bringing us back to the model originally proposed by Sokransky and colleagues in 1998 that is also known, and they were really clever in 98. They named this the microbial complexes theory. And this is really nothing more than a contraction of the word microbiome. So microbial complexes theory is really the microbiome, where the organization of the bacteria in the community and the role of individual species within that community can impact on the development progression of, here, in this case, periodontal disease. But as that diagram categorizes, the bacteria and these color-coded complexes of the Sokransky complex can lead to our understanding of plaque formation and disease progression.
So it's not just a gamish.
No.
They're actually defining particular interactions and what adhesins mediate those interactions that are one key step in building of a biofilm. So I would imagine that this work would be important or of interest to toothpaste designers, right?
Well, in the early days of toothpaste, what they would put in toothpaste, and this was a very successful toothpaste, is triclosan, which is a surfactant that just disrupts communities, and it's been banned. So we lost a great toothpaste because...
No, we did not.
Well, from the perspective of disrupting...
problems.
From the perception of disrupting a biofilm.
I think the mechanical action of brushing your teeth is pretty good at disrupting.
Oh, no,
them.
that's fantastic. We've known that since we were in the woods with the sticks.
Yeah. All right. How about a mouthwash then? That has inhibitors of some of these interactions.
That would be great.
I mean, could you put in a B. parvella that's already attached to this is where actually genetically, because I'm looking at figure 7 and it looks like you could put in a modified version of this that could outcompete or essentially doesn't stick.
So like a probiotic cause toothpaste.
Yeah.
You guys have figured it out.
I mean, but the problem really is the lactic acid that the thing that's above this in the biofilm is producing, right?
Well, no, it's below. It's
below it, right? Sorry, below, above, yeah. But that's the big problem.
You need the lactic acid to feed the Vionella. That's the only thing it eats.
But the lactic acid is what's causing the cavities, correct?
Right. And you want your pH to be as close to 7 continuously. And when we eat a meal,
they'll stop there. Okay, so they need it to keep it. Okay, so the biofilm balances out the pH again. Anyways, I do really love figure 7. It's very pretty.
Yeah, figure 7 sums it up beautifully. And the transcription models that they got in there, you need to commit. They're beautiful figures, and they really drive home what they found.
Well, this is the work of Louis Dorison, who grew up in Paris, living just 5 minutes away from the Pasteur Institute, which was founded in 1887. It's a nonprofit foundation that is dedicated to prevention and treatment of infectious disease. And his great aunt was a researcher there. So at a very young age, he was fascinated by microbiology. He knew that's what he wanted to study. So he was fortunate to go to a school in Paris, École Normale Supérieure, a top French university that has very few students, but their mission basically is to prepare these people to go on and be educators and researchers. So as a high schooler, he was able to go to a number of international labs and work on synthetic biology at the Institut Pasteur, microbial symbiosis in corals and anemones in Melbourne, RNA modifications at the Max Planck Institute in Berlin, and then also the microbiota at the INRAE, which is an institute also in Paris. So all of this was the experience as a high schooler.
Oh,
as a high schooler.
Oh, my.
So then, no. At the university, I'm sorry.
Yeah, at the École Normale Supérieure.
So he then got a master's at the Institut Pasteur, and he did an internship with Christophe Boulogne, who's one of the authors here, one of the senior authors, and then continued in that lab, Christophe's lab, for his PhD thesis. And he's currently staying for another 6 months to finish up some work, but has got a postdoc lined up at the Douve Institute in Brussels with Geraldine Lelot. He'll be working on Delavibrio.
Delavibrio. Oh, that's so cool.
Mark Martin's favorite organism.
Yeah.
It's a really nice lab.
That's great. He's hoping to be able to come back to France and be a researcher, hopefully lead his own group. He also wants to point out that early in his training, he and his family realized that he had his hands. He wasn't able to do a lot of writing by hand, and it was getting in the way of his education. So he needed to use a computer starting in middle school for taking notes in class. And some of the administration of the schools had a problem with this, thought it was an unfair advantage. So both he and his parents had to really stand up and make the case. And he wants to say that without the school agreeing to be inclusive and accepting that this is a tool that he needed to use, he said he wouldn't be in the position where he is today. So he wanted to point that out because he's aware of the DEI attacks that are currently underway here in our country. He also wants to thank that not only was Christophe an amazing thesis supervisor who gave him freedom and also advice that he needed, he also wanted to thank his colleagues Mark Herzberg and Karen Johnson, who shared their bank of Streptococcus gordonii mutants, which were essential for this. And then Justin Merritt also shared the tools that he uses to modify that Streptococcus species. So he really has had the experience of science being a very open field, people sharing tools, strains, plasmids, ideas that really enriched his experience. He did point out that he got that library of the Streptococcus mutants just as he was finishing his master's internship and had to just put them into the -80 and store them away while he waited 4 months for the beginning of his PhD, also with Christoph there at the Pasteur Institute. And he said it was just painful to have to wait those 4 months because he couldn't wait to
burning a hole in his
pull
pocket
out of those mutants. Yeah, and also AlphaFold, which we talked about earlier, came out during his PhD, and it was a really exciting day when he was able to model for the first time this adhesion that had been working on for more than a year. So in reflecting on his own experience, he thinks junior colleagues realize that when you're choosing a lab, it's really important the relationship you have with your thesis advisor. He says you need to find a place where you're valued, given the freedom you need, but at the same time, given the support you need, able to go talk to your advisor and run some issues, some ideas past them. So he says, reflecting, he knows that that PhD training can be mentally difficult. So it's good to be surrounded by people that will support you and make it easier. And in general, he said, as to be a scientist, you've got to learn to be resilient with failure. Some experiments aren't going to work. Some PCR reactions aren't going to work, but realize it's not your fault. It's just the process of science. And for the few times that it does work, it's all worth it. And I would agree and say, Louis, that one of my favorite expressions is there's a reason we call it research and not search.
Absolutely, Michelle. And the other thing is his experiences actually probably greatly helped him in these studies. And, life is cumulative. And all of those opportunities he had as a young person really paid off in helping him develop this clever thinking that he has and being able to dissect this very complicated problem that we've understood. Look at Sokransky, he published that in 98, and we're only beginning to work it out now.
I'd like to add that more than 20 years ago, I was fortunate to go to the Pasteur Institute for a sabbatical. I was there for a full year when my kids were were like 12 and 14 years old. And I worked with a Legionella biologist, Carmen Buchreiser, a group leader. But I was also interested in Legionella. It's an environmental microbe, and we knew it formed biofilms in engineered water systems. Jean-Marc Guigaud, who's one of the senior authors, and also Christophe Boulogne were really gracious and welcoming my naive presence at their lab meetings and allowed me to kind of mess around and do a few experiments. So it is a wonderful place to do research, and in particular, that group of researchers is really talented and welcoming.
Thank you, Michelle. Thank you, Michael. That's TWiM 330. You can find the show notes at microbe.tv/TWiM. Please consider supporting us. We need your help to do these programs. Microbe.tv/contribute. Michelle Swanson's at the University of Michigan. Thank you, Michelle.
Thank you.
Michael Schmitz at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone.
And Petra Levins at Washington University. Thanks, Petra.
Thank you.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiM, Ronald Jenkins for the music, and editing by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.